Newly synthesized MHC class 1 molecules are retained in the endoplasmic reticulum until they bind peptide. Binding of peptide is an important step in the assembly of stable MHC class 1 molecules. When the supply of peptides into the endoplasmic reticulum is disrupted, as in TAP mutant cells, newly synthesized MHC class 1 molecules are held in the endoplasmic reticulum in a partially folded state. This explains why cells with mutations in TAP1 or TAP2 fail to express MHC class 1 molecules at the cell surface. The folding and assembly of a complete MHC class 1 molecule depends on the association of the MHC class 1 alpha chain first with beta 2 microglobulin and then with peptide. And this process involves a number of accessory proteins with a chaperone-like function. Only after peptide has bound is the MHC class 1 molecule released from the endoplasmic reticulum and allowed to reach the cell surface. In humans, newly synthesized MHC class 1 alpha chains that enter the endoplasmic reticulum membranes bind to a chaperone protein, calnexin, which retains the MHC class 1 molecule in a partially folded state in the endoplasmic reticulum. Calnexin also associates with partially folded T cell receptors, immunoglobulins, and MHC class 2 molecules, and so has a central role in the assembly of many immunological proteins. When beta 2 microglobulin dissociates from calnexin and now binds to a complex of proteins, one of which, calreticulin, is similar to calnexin and probably carries out a similar chaperone function. A second component of the complex is the TAP-associated protein capucin, also encoded by a gene that lies within the MHC. Capucin forms a bridge between MHC class 1 molecules and TAP1 and TAP2, allowing the partially folded alpha beta 2 microglobulin heterodimer to await the transport of a suitable peptide from the cytosol. A third component of this complex is the chaperone molecule ERP57, a protein disulfide isomerase that may have a role in breaking and reforming the disulfide bond in the MHC class 1 alpha 2 domain during peptide loading. ERP57 and calreticulin bind to a number of glycoproteins during their assembly in the endoplasmic reticulum and seem to be part of the cell's general quality control mechanism. Finally, the binding of a peptide to the partially folded heterodimer releases it from the complex of TAP, capucin, calreticulin, ERP57. The fully folded MHC class 1 molecule and its bound peptide are now able to leave the endoplasmic reticulum and are transported to the cell surface. And that's newly synthesized MHC class 1 molecules are retained in the endoplasmic reticulum until they bind peptide. Have a great day.